Hey everybody, welcome back to another lecture. Happy Wednesday. Today we're going to be doing our enzymes, or first look at our enzyme. Kinetics and inhibition. Now, if there's a magical component to life, an argument can surely be made for it being catalysis, right? Catalysis being the job that enzymes do. Thanks to catalysis, reactions that can take hundreds of years, uncatalyzed, will take place in seconds. Chemical catalysts like platinum speed up reactions. If you remember from organic chemistry, right, platinum catalysts are used in reduction reactions. But chemical catalysts can't even touch biological catalysts. Enzymes put chemical catalysts to shame. So to understand enzyme catalysis, we first have to understand a little bit about energy. Chemical reactions follow the universal trend of moving towards lower energy. And we call this, we usually refer to this as the delta G. And we want delta G, well, when delta G is negative, reactions are going to be spontaneous. Now that doesn't mean they're going to be fast, and that's where enzymes kick in. Now the reason that enzymes can be much more catalytic than your typical chemical catalysts is the mechanism of action. And we're going to spend a little bit of time talking about that. Okay, so enzymes are biological catalysts. They're usually globular proteins, with the exception of some self-splicing RNAs. Before there were, if you remember from reading chapter 1, before there were proteins which get made by, RNA and DNA, there were no proteins, right? Before, proteins themselves, of course, get made by RNA, so, and they were, that reaction's already being catalyzed. So if they weren't around, who was catalyzing? Well, it was the RNA. So there are some RNAs that do catalysis. Increasing reaction rate by a factor of 10 to the 20. Very specific to the extent that they can distinguish stereoisomers of a given compound. Very finely tuned by regulatory processes, and we're going to talk about those. All right, so to start, let's talk about the standard free energy change, or delta G. And delta G is the difference between energies of the reactants and the products under standard conditions. Now, enzymes will speed up a reaction, but they cannot alter the equilibrium constant or change the free energy. Okay. The reaction rate will depend on activation energy, and this is usually known as delta G with this little symbol that represents, or delta G naught, with this little symbol that represents a transition state. So delta G with this little symbol is for an uncatalyzed reaction. For an uncatalyzed reaction is higher than that for a catalyzed reaction. So the amount of energy that it takes, right, the activation energy is higher. And so you can see here in these two diagrams, they roughly start at about the same energy. The products have just about the same energy. The only difference is that the activation energy, the hump, has been reduced. So instead of having to go over this big hump, it only has to go over this little hump. Okay. Now, the intermediate stage in a reaction, this little transition state up here, this is where old bonds have to break, new bonds have to form. And so you can see that that's the basically the hump that molecules have to overcome. Now, when we plot these, let's see. When we plot these, what we see is that enzymes somehow overcome this energy hump. And the way that they do it often is by holding substrates in the correct position, sometimes bringing them close together. And that has a large effect on making the reaction actually happen faster. Now, so again, the secret to the catalysis is the way that the activation energy is lowered. So let's talk a little bit about that. So enzymatically catalyzed reactions are orders of magnitude faster than uncatalyzed reactions. And chemical catalyzed reactions don't have all the tricks or the tools that proteins are able to use. So one area in which they're the same, and something that probably every chemistry student has heard or been told, is that a catalyst speeds up a reaction without being consumed. So it ends up just the way it was before the reaction. So enzymes share that property, but in the middle, during the actual catalytic action, the enzyme is in a transient state, a transition state. And it's the ability of the enzyme to change like that that leads to leads to it being incredibly efficient. So these changes may be subtle electronic ones, more more significantly covalent modifications or structural changes that arise and just from the flexibility inherent in enzymes that are not present in chemical catalysts. Ability for double bonds to move around, or for a molecule or side groups to be ionized or possibly reduced or oxidized. So this flexibility allows for the movement, and movement facilitates the alteration of electronic environments necessary for this catalysis. So enzymes are much more efficient than the rigid chemical catalysts in this respect. Now, as far as substrate binding, another important difference is that the enzyme can bind or like grab a molecule and put it in position to be sort of electronically induced to react with itself, with another substrate. Chemical catalysts can't do this. Enzymes have an active site. We're going to talk a little bit more about active sites. But substrates bind an active site and are oriented to provide access to the relevant portions of the molecule and the electronic environments where reactions are more conducive. And then, of course, I mentioned that they are very flexible. All right, so let's see. So let's take a typical reaction. Consider this reaction. So we've got hydrogen peroxide into water and oxygen. So platinum catalyst will actually increase the rate of this reaction. And so you can see relative rate here about 2.7 10 to the 4 versus no catalyst, right? This will happen on its own, but relative to that, it'll happen 27,000 times faster with a platinum catalyst. But with biological catalyst, you see the big difference there. Now, there are some other things that affect catalysis and enzyme rate, and so let's talk a little bit about that as well. Now, we usually calculate delta G for reactions taking place at a constant temperature. You may have seen some of these reactions, or these equations. Delta G naught is equal related to enthalpy and the negative of the change in entropy. We can even relate delta G to initial and final states of a reaction by including the equilibrium constant. We get delta G naught is the negative of this this constant times the temperature in Kelvin, natural log of the equilibrium constant. So we have ways of finding out changes of free energy. But you see here temperature is included. So temperature definitely has a component. And there is a temperature dependence on catalysis. And so I've got actually that right here. You can see that increasing the temperature of a reaction will actually increase the available energy, not just to the solution and the temperature, but to all the reactants, so that they can reach those transition states a little easier. So it also lowers activation energy. Now this, with biological reactions, only works to a limited extent, because as we saw earlier in proteins, if we increase the temperature too much, we might get enzyme denaturation, and that's not what we're going for. All right, let's see. Let's talk a little bit about enzyme kinetics. We need to talk about kinetics. So let's take a typical reaction. We could use the same one I just had up there, but I want to go simpler. Let's say A plus B gives us P. And this is the reaction, and so we want to talk about the rate. So the rate of the reaction can be expressed in a few different terms. Now we can talk about the rate of the reaction in terms of the disappearance of one of the reactants. So the rate of the disappearance of A, for example, would be the negative change of the concentration of A per some unit time. So it would go down by such and such amount of A per second, right? That would be a rate. That's equal to rate. There's a few other ways, right? We could do it for B. The negative rate of change, or the negative change of the concentration of B per unit time is also rate. But we could also say that it's the rate of appearance of P, right? So that could also be equal to the positive change in product per unit time. All of these should be equal to each other, okay, in terms of calculating the rate of one particular reaction. Okay, so we could say then that they're all equal to each other. We could say that the rate, or it would be equal to, and I'm not going to write both of these out, but the rate of the negative change of A per time would equal the rate of change for P per unit time. These would be the same, and also for negative B. Okay, now we can include a proportionality constant, okay? And we call these proportionality constants. We give it the value K, and so we would include in a rate expression, right? Because at the moment, rate is proportional to the concentration of A raised to some subscript depending on, we experimentally determine these numbers. We call them the order. And concentration of B also to some other number. And so we would write a rate expression as being something like this, where N and M both get experimentally determined, and K would be a rate constant for that given equation. And so these things have to be figured out by doing some experiments. Now, an overall reaction is the sum of all the total exponents. So, in the rate of the reaction of A to P, we would only show one type of rate equation. Like if we're looking at how much A turns into P, we'd say that the rate is equal to the reaction constant, or the rate constant, and let's say that the exponent is 1 here, okay? This would be a first order reaction because of this little exponent here. So first order is a reaction whose rate depends on the first power of the concentration of the single reactant. So if I were to double this, it would double the rate. If I were to triple the concentration of A, it would triple the rate. These things are related to each other in that way. Now let's say that I have another reaction. Let's say I have a reaction that's A plus B, so this one requires 2 substrates. And they turn into, it could turn into P, it could turn into C and D. It doesn't really matter. Let's put P here. So the rate of this reaction would be the rate constant for this reaction, concentration of A, let's say concentration of B as well, and have something like that. Let's say that we've experimentally determined both of these exponents are 1s. And so this would be first order with respect to A. It would also be first order with respect to B, meaning that if I doubled the concentration of B, the rate would go up. If I doubled the concentration of A, of course keeping B the same and keeping A the same, when I do it for B, the rate would double. If this was a second order reaction in respect to B, then by doubling B's concentration, I'd actually quadruple the reaction rate, because it would be related to the exponent. It would be 4 times as much, or to the square. Maybe what I just said about the square bit was wrong, but if I were to double B, it would quadruple the rate. And so to get the overall order, we would say this is first order respect to A. This is second order with respect to B, so it's overall a third order reaction. Or in the previous example, when this was a 1, it would have been a second order reaction. So that's what these terms mean, okay? I can give you another example of a second order reaction. So we've got glycogen. Now, glycogen with some number of sugar molecules on it, inorganic phosphate, and these guys will react together to produce glucose 1-phosphate, and then, ran out of room, glycogen, N-1, so one less sugar unit on it. So this would be an example of a first order reaction with respect to glycogen, a first order reaction with respect to inorganic phosphate, and a second order reaction overall. We could say that the rate of this reaction looks something like that. Okay. We can also talk about zero order reactions. And so, like, let's just say I have A going into P and that the rate is K, and that this is a zero order reaction. Anything raised to the zero power is just a 1, and so that basically means that the rate is just equal to K. It's irrespective of the concentration. I don't know if that's the right word. And so even if I tripled A, the rate wouldn't go up. So these are the different type of ordered reactions that we have. What's on my next slide? Lock and key. We're getting here. I need more room though, so I'm going to stay here. In the enzyme catalyzed reaction, there are a couple terms. There's the substrate. We can go to the next slide. There's a substrate, usually designated S. Then there's the active site. And so you can see these guys here. Substrate, active site. That's the portion of the enzyme surface to which the substrate binds. This can be via non-covalent forces. It usually is. And so van der Waals interactions, hydrogen bonding, electrostatic attractions. First step in the binding, it's the first step in the binding of, what I'm trying to say is the first step of catalysis is the binding of enzyme to substrate. That has to happen first. Now there's a few different ways in which this is thought to happen. There's the lock and key model, which basically says that every substrate is like a specific match for every active site of an enzyme, or for an active site of its enzyme. And then there's the induced fit model, where the binding of the substrate actually induces a change in the conformation of the enzyme that results in a more complementary fit. And the binding site would have a different shape before it's bound, and it would have a different shape after it's bound. This induced fit model actually seems to match more with what we see in nature. For example, enzymes can alternate between T states, or tight states, which have low activity, and R states, which have relaxed, or R for relaxed, which have a greater activity. The induced fit model seems to match what we see for enzymes that have more than one substrate that they can bind to. Let's see, what else about the induced fit model? We can talk a little bit about the different types of reactions that there are in terms of enzyme binding. There's single substrate, where, and like let's say a substrate can be converted into a product. There can be a single substrate being converted into a couple products. There can be multiple substrates being turned into a single product. There can be multiple substrates that get converted into new products. And of course, all of these would have reversible directions. One thing to mention about equilibrium, I don't know if you guys know this, because students usually are generally confused about some of this. The equa part in equilibrium does not mean absolute concentrations. What happens in a biochemical reaction is that at equilibrium, the concentrations of reactants and products don't change over time. The reaction didn't stop. The forward and backwards directions are happening. It's just that concentrations of the two amounts don't change. Now, that doesn't mean that the concentrations are the same. They may not be, and right, that depends on the equilibrium expression, on whether or not the KEQ is bigger than 1, less than 1, and how much bigger or less than 1 it actually is. Let's see. What's my next slide here? Okay. So let's talk about the process of enzyme binding. So it goes through this equation sort of down here at the bottom. I mean, you could actually simplify it a little bit and just say that E plus S can actually react together to form product, right? Enzyme and substrate bind and make product. Now, there's also sort of a more expanded way that we can look at it that includes the enzyme substrate complex. So let me, oops, that's not what I want to do. Enzyme substrate complex. So ES forms, E and S form an ES, and then this, while it could just form EP, there's actually a more expanded version of this as well, where we can say that ES form an ES activated state, and that this ES activated state will then bind or will then convert into an enzyme product complex and then release product. So there's a few different versions of how we can look at this. And so you'll notice that enzyme substrate complex is a little lower in energy right off the bat than the initial amount, and that's to help this process, start, help get it to form. We're going to need then to overcome that activation energy, and this is the part where the enzyme does that good job. And then where the proximity and the orientation help, of the molecules to each other help speed up the reaction, and then they release their product. Now, there's a few different mechanisms that are thought to proceed here. We can talk a little bit about these as well. The first mechanism is the ordered mechanism. This is where, let's say, we have enzyme, and then enzyme will react with a substrate to form enzyme and some, enzyme, let's just call it a EA complex, and then let's say this was a 2-molecule reaction, then B would come in, and we'd have the enzyme complex with the A in it, and then these guys would react, forming, let's just give it some new letters, P, enzyme, and Q, and then it would release, let's say P, leaving behind enzyme and Q, and then we get enzyme releasing Q. This is called ordered mechanism. As opposed to an ordered mechanism, we may have a random mechanism, where let's say maybe A and E can form an enzyme E complex or enzyme A complex, which can then react with, well, we could also have A though, or enzyme and B forming first, forming the enzyme B complex. They can both then accept A or then accept B to form the A-B enzyme complex. And then just like what we saw, they can disassociate in different orders as well. We can get this P enzyme Q complex dissociating into, let's say, Q and E first, or maybe E and P first, and so on and so on. This would be a random mechanism. And then there's actually one more. There's a ping pong mechanism, and the ping pong mechanism is a little different. This one would have, let's say, enzyme and substrate A reacting to form enzyme A, and then this reacts to form enzyme and some version of a product, which can then actually be ejected, leaving behind an enzyme molecule ready for the next substrate to come in. Let's just say, B, and then enzyme primed enzyme and B can then react to form enzyme and finished product Q, and then let's say enzyme releases Q. So here the enzyme first releases some part of it, some part of a substrate molecule, some part of A, and then later finds B. This would be ping pong mechanism. The other two would be sort of concerted, happening at the same time, either through ordered or through random mechanism. This one would be sort of different than that. Okay. Let's switch gears. Let's talk about kinetics. Now this usually scares people, and of course I can't spell kinetics, so I'm going to go ahead and backtrack on that one. So kinetics is K. There we go. So to understand how enzyme kinetics or enzymes enhance the rate of a reaction, we have to understand kinetics. So we're going to talk about a model here called the Michaelis-Menten model. So in order to understand the model, we have to understand a few parameters. So first, we'll describe a simple reaction in terms of E plus S, apologize for my poor E, can react and dissociate together into ES complex, which can then form, we're going to say, ES activated state, enzyme product complex, and then enzyme and product. All of these, well, the first step is the reversible step. Once we get to this enzyme activated state, they're all basically forward directions. Okay. So the first consideration we have is velocity. Okay. The velocity of a reaction is the rate of creation of product over time, measured as the concentration of product per time. So the time is a critical consideration when measuring velocity. In a closed system in which an enzyme usually operates, all reactions advance towards equilibrium. Enzyme catalyzed reactions are no different. And the end result from a enzyme catalyzed or a non-enzyme catalyzed reaction is equilibrium. Enzyme catalyzed ones get there faster. Equilibrium, the ratio of product to reactant doesn't change. This is a property of equilibrium. Since the system's closed, the concentration of product over time doesn't change. The velocity will thus be zero under these conditions, and we will have learned nothing about the reaction if we wait too long. So we don't want to look at reactions at equilibrium. Okay. So consequently, the Michaelis-Menten kinetics equations or model, velocity is measured as initial velocity. And this is accomplished by measuring the rate of formation of product really early in the reaction before equilibrium is established. And under these conditions, there is very little, if any, of the reverse reaction happening. The other two assumptions that we're going to have to make are related. First, we use conditions where there is much more substrate than enzyme. This makes sense. If the substrate is not in great excess, then the enzyme's conversion of substrate to product will occur much faster than the enzyme actually binding substrate. And so if it's having to wait for substrate, then there's not, if there isn't enough substrate, then it's having to wait. And then we're not getting a good, accurate rate, okay? Because we're waiting too much time for the enzyme to bind the substrate. So we assume saturation of the enzyme will be when we put in an abundance of substrate. We assume that the enzyme will be saturated, and as such will give a maximum velocity of the reaction. So a couple of graphs here. So first one you can see is just the concentration of product, substrate, and enzyme, and even the enzyme substrate complex just versus time for an enzymatic reaction. So initially you can see P is very, very low, right? And over the course of the reaction, the product gets higher. Substrate starts off very, very high. By the end of the reaction, the substrate is very low. Enzyme substrate complex at the beginning is also low. It goes through a peak, and then as the substrate concentration goes down, the enzyme substrate concentration also goes down. Now, free enzyme, we see, starts off relatively high. It goes down, and that makes sense. When, there's the lowest amount of free enzyme, we see the highest amount of bound enzyme. And then as bound enzyme is going down, we see lots of free enzyme. So this is just a typical look at how these reactions sort of play out. All right. Oh wait, I thought I had another. So let's talk a little bit about steady state. So we get a the high concentration of substrate combined with measuring the initial conditions results in basically studying reactions that are so-called steady state reactions. And so the steady state reactions typically take place right here at the beginning in this little time frame. Okay. This would be pre-steady state. And this is where the enzyme and the enzyme substrate complex are not changing very rapidly. So these are the conditions that we use. We are measuring the initial velocity, substrate is in great excess, and the reaction conditions are occurring under steady state, which is that the concentrations of those enzyme substrate complexes don't significantly change that much during that time. Okay, let's take a look a little bit at how studies of kinetic properties of enzymes are actually done, how they're actually conducted. So to perform an analysis, one would do the following experiment. You take 20 test tubes, set up with an enzyme buffer to keep the enzyme stable. Same amount of enzyme would be put into different amounts, and then different amounts of substrate would go into each tube, ranging from tiny amounts in the first tubes to large amounts in the last tubes. And the reaction would be allowed to proceed for a fixed, short amount of time, and then the reaction would be stopped, and the amount of product would be determined. Now, the initial velocity, V0, would be very, very low, and the initial velocity would be the concentration of product found in each tube divided by the time that the allowed reaction was allowed to run. So it's the delta P concentration per whatever unit, and we're going to call this the initial velocity, or V0, my bad, V0 goes down here. And then we would plot this. We would plot the data. We would put V0 on the x-axis. We'd put the concentration of the substrate on the y-axis. V0 on the y-axis, concentration substrate on the x-axis. You guys can see that. And then, of course, each tube having a different concentration. We could plot it and we could get some kind of result. And so what you can see from both of these graphs, which are relatively the same, is that we get a curve that actually shows an enzyme that's following Michaelis-Menten kinetics, in that at low concentration of substrate, it's binding it very, very quickly. It's converting it into product as soon as it can. At low concentrations, we see that the rate of increase of product is almost linear with the amount of substrate. So we call this the linear initial accumulation. Now, what eventually happens, though, is that we get to a nonlinear increase. So as the substrate concentration increases, the velocity of the reaction actually ceases to increase linearly and instead starts to flatten out. We see it more in this part, and then so we're gonna look at this line that it's kind of approaching, that never actually reaches. So it flattens out, basically indicating that as the substrate concentration gets higher and higher, the enzyme has a harder and harder time keeping up with converting that into product. So saturation is what we're talking about. So when the enzyme becomes completely saturated with substrate, it doesn't have to wait for substrate to diffuse into it, and will be operating essentially at its maximum velocity. Every time it spits out a product, it grabs another one. And so this is the limit of basically how much product you have. So for an enzyme that's following Michaelis-Menten kinetics, we can actually measure the velocity of the reaction at any time if we know what Vmax is, if we know the substrate concentration, if we know what Km is, and if we, again, know that substrate concentration, we can determine this. So for these two terms, we need to talk about them. We haven't explained them. So the first term is Vmax. Now Vmax refers to the maximum velocity of an enzymatic reaction. Maximum velocity for the reaction occurs when an enzyme is saturated with substrate. Saturation is important because it means that none of the enzyme molecules are waiting for substrate. So saturation ensures that another substrate is waiting instantly, and the units of Vmax are concentration units per time. So let's just, molarity is a general concentration term, per time. These are the units of Vmax. And so on this plot, you can see the initial velocity versus substrate concentration. Vmax is the value on the y-axis that the curve asymptotically approaches. I don't know if that's the right word. You know what I'm trying to say. It should be noted that the value of Vmax depends on the amount of enzyme used in a reaction. If you double the amount of enzyme used, you double the Vmax. So if you wanted to compare velocities of different enzymes, you'd want to use the same amounts of the enzymes. All right, let's talk about Km. Km is the second term. Km refers to, or is referred to as the Michaelis constant. It is a substrate concentration that causes the enzyme to work at half of the maximum velocity, so a Vmax over 2, or 1/2 Vmax, as people say. It measures, in simple terms, the affinity for the enzyme and the substrate. So if you have a really, let's see, how do I want to say this? Hold on, I wanted to sum it up in a nice way, but I forgot how I wanted to say it. It's inversely related. So the bigger the Km, the smaller the affinity, the smaller the Km, the larger the affinity. That's what I was basically trying to go for. So what it means is that enzyme or substrates that have low Kms have high affinity, and it takes less of that substrate to get to Vmax. They bind really, really nicely to the enzyme. It's not always the case. Some enzymes and substrates have really high Km values and lower affinity, and it takes much more of that substrate to get to the value of 1/2, or to get to the 1/2 of the maximum velocity. Affinities of enzymes for substrates varies considerably, so knowing the Km actually helps us to understand how well an enzyme is suited to the substrate being used. So, and of course measurements of Km depend on measurements of Vmax. I wanted to give you the units of Km, which is essentially, what is the unit of Km? Well, it's a concentration unit. That's all it is. Km is just a concentration unit. And actually, that's a common mistake that students make, is that when they're talking about what Km is, or describing Vmax even, they say that Km is Vmax over 2. Of course, this isn't true, right? Km is a substrate concentration, and Vmax is a velocity, and those two things don't jive. So while we know what Km means, it means it's 1/2 of the maximum velocity. They are definitely not related to each other more than that. Let's talk about kcat. There's one other term that we like to use. Actually, I could probably talk about kcat here. So kcat. So kcat is actually the Vmax of an enzyme divided by the concentration of that enzyme. Now you're wondering, did I spell it right? Why on earth do we do this? So it's desirable to have a measure of velocity that's independent of enzyme concentration. Remember, if we double enzyme concentration, we double Vmax. So we use kcat for this. Sometimes it's also known as the turnover number. The kcat number, first you have to determine a Vmax, and then you divide it from, you divide the enzyme concentration from that, and that gives you the kcat. Now, since KCAT is essentially, let's talk about what the units are. Since Vmax has a unit of concentrations per time, and enzyme is a unit of concentration, the units of KCAT are per second. So just to the seconds to the -1. So that seems a little unintuitive, but it means that the value of KCAT is the number of molecules of product made by that enzyme molecule per second. So a KCAT value of 1,000 per second means that each enzyme molecule in the reaction at Vmax is producing 1,000 molecules of product. So since KCAT is a calculated value, it can't be read from a velocity versus substrate concentration graph like Vmax and Km can, but we can calculate it. Now I've shown you a couple here, so you can see that there are some enzymes that have really big KCAT values. So a KCAT value of 1,000 molecules per product, per enzyme per second seems high, but there are enzymes like you can see here, carbonic anhydrase, with over 600,000 molecules of product per second. I mean, that's astonishing. This is why I said the catalysis is magical. So in contrast to Vmax, which varies with the amount of enzyme used, this is a constant number, and it's also specific for a specific enzyme. So every enzyme is going to have its own KCAT in its own environment. Now, not every enzyme follows Michaelis-Menten kinetics, so we have to mention that here. Many enzymes have multiple protein subunits, and these can interact differently. We're going to look at an example of that by the end of this lecture. Now, we also like to talk about perfect enzymes. If you think about what an ideal enzyme might be, it would be one that has a very high velocity, a very high affinity for its substrate, right? So it wouldn't take a lot of substrate to get to half of Vmax, and the KCAT would be very high. These enzymes exist. There are enzymes that have values of KCAT and Km that are maximum. And when we look at the properties of these enzymes, kind of compare them to each other, we see that they all kind of have approximately the same value. So it's almost like being perfect really is a limit or an attainable thing here in our bodies. And so these are them. So when you look at these perfect enzymes here, you see that carbonic anhydrase is definitely on the list. I pointed to the right one there, or wrong one there. But all of these enzymes have they're all referred to as perfect because they've reached the maximum possible values. All right, and there on the side is triose phosphate isomerase from Wikipedia. Just thought I'd include a perfect enzyme so we can look at it. All right, this is what triose phosphate isomerase actually does. You might wonder, what's the point of having a perfect enzyme? Is it just a fluke? Actually not. This enzyme catalyzes the conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. This is I think this is step 5 in the glycolysis reaction. Now, why does this have to happen? Well, if we don't have dihydroxyacetone phosphate converting into glyceraldehyde 3-phosphate very quickly, there is an intermediate, this enediol intermediate, that can turn into methyl glyoxyl, and this guy is not good for our bodies. And so this enzyme almost had to evolve this speed to prevent this side reaction. If we do this reaction in glassware, in a test tube, in a beaker, we get methyl glyoxyl without the enzyme. With the enzyme, none of this happens because it's all going back and forth this way. So again, nature has found a way. All right, we need to talk about some Lineweaver-Burke problems here, or Lineweaver-Burke plots. All right. So the study of kinetics, and you guys we didn't do very much math here, but I encourage you to look in the book and to, calculate Km and Vmax from a plot. Take some of that sample data in the back of the book, in the back of one of those chapters, and put it in Excel and just enter substrate concentration and velocity data and see what Vmax is for an enzyme. I mean, it's worth doing once, and then, if you're not going to go into this stuff, you don't have to do it ever again. But this is, and we haven't even gotten to what I'm going to mention, but this is the most math-intensive component of biochem, the Lineweaver-Burks and the enzyme kinetics. It's one of the most daunting aspects of the subject for students. Some attempts are made to simplify the math. Sometimes that just frustrates and confuses students. And often one of the culprits of the frustration and confusion is these Lineweaver-Burk plots. So I often just say, hey, memorize these. I mean, and if you've gone over these before and you've been told, just memorize them. I mean, who can blame you for being confused? Both of these plots that we're going to look at are basically aimed at simplifying the determination of Km and Vmax. That's it. They're trying to make determining Km and Vmax easier. In making these plots, all they're doing, I mean, they're taking the same velocity times, or the same exact substrate and velocity data, and they're just making it easier to plot. They're manipulating it. This is done by using a double reciprocal. So instead of velocity, they're going to use 1 over velocity. Instead of substrate concentration, it's 1 over substrate concentration. By inverting these things and doing both of them double reciprocal, we see that this actually plots as a straight line, essentially, and we can get a y-intercept that correlates to 1 over Vmax. We can get an x-intercept that correlates to 1 over Km, and we're able to get Km and Vmax from these. That's it. That's what they're for. That's the point of these equations and these plots. We're going to come back and use these as ways to investigate what type of inhibition may be working on our enzyme, because we're going to see that different types of inhibitors tend to affect these plots. And so we'll want to compare a non-inhibited plot with an inhibited plot and see what changes. If Vmax changes, it means one thing. If Km changes, it means another. If they both change, it means a third. So that's what we're going to use these for. All right. So again, a couple turnover numbers and some Kms for some typical enzymes here. All right. Let's talk about some specific reactions now. We're going to look at 2 enzymes really fast. We'll go into it more in the next lecture, but we'll look at these 2 enzymes really quickly and just do some quick Michaelis-Menten plots on them and see what we get. So the first one we're going to look at, and of course we're going to go into the mechanism of action on chymotrypsin a little later in the next lecture. But chymotrypsin catalyzes the hydrolysis of peptide bonds, where the carboxyl carbon is actually, where this thing, it slices off aromatic residues. So if it finds amino acids that have aromatic residues, it tends to go and cut them. Now, so phenylalanine, for example, tyrosine, these are culprits. It also hydrolyzes ester bonds, and so you can see an example of that here. So I guess I should say this guy would have to be a phenylalanine, or it would be like I said, a tyrosine, something aromatic. Or ester bonds. Ester bonds are similar to peptide bonds in that there's a carbonyl, and then there's a nitrogen or an oxygen here that can either then become an amine or an alcohol. But we're gonna get back the carboxyl side of the peptide or of the molecule. So this is how the reaction, or the types of reaction that chymotrypsin can actually go after. Now, specifically, let's take a look at a system that we can use to monitor the enzyme that catalyzes this reaction, so chymotrypsin. So if we take p-nitrophenylacetate, you can see here that if we hydrolyze that ester bond, we end up with a product with a different color. So we can monitor the appearance of this product. It makes it a good system to study. Okay. And so we're going to look at how fast the product We're going to do a velocity versus substrate concentration plot on chymotrypsin, and we're going to compare that to a different enzyme. So now we're switching gears. So we're going to talk now a little bit about this enzyme. This enzyme is called aspartate transcarbamylase. You can kind of see all the parts. Aspartase, transcarbamylase, aspartate, sorry. We call it ATCase for short. Aspartate transcarbamylase. This is an allosteric enzyme, so we haven't talked about that. This is an allosteric enzyme that catalyzes an early reaction in pyrimidine biosynthesis, and here's kind of a version of that. You can see some of this reaction. Now, I told you we're going to compare two plots. Here they are. So here is chymotrypsin, velocity versus substrate concentration, substrate in this case being the p-nitrophenyl acetate. And then here, ATCase, substrate being aspartate. And they look completely different, right? The one of them is a sigmoidal curve. That's what we call this here. This is a hyperbolic. This is sigmoidal. So this indicates that there are two, at the moment for us, two types of enzymes. There are enzymes that are what we can call regular enzymes, and then there are these allosteric enzymes. They are under some other kind of regulation or control, and really the subject of the next lecture that we're going to do. We're going to look at more of this particular ATCase, but we're going to look at hemoglobin as well, these allosteric versions. And then we're actually going to look at how chymotrypsin works too, but that's coming. Okay, let's talk about inhibitors. So hold on to that. So yeah, ignore all of this up here. It should just say E plus S gives us ES, or E plus I gives us EI. I don't know what happened. So inhibitors are substances that decrease the rate of enzymatic catalyzed enzymatically catalyzed reactions. There are reversible inhibitors that can bind and unbind. They can be competitive or non-competitive, even uncompetitive. And then there are irreversible inhibitors. And these are substances that react with enzymes to produce proteins that are not enzymatically active anymore. And no more original enzyme can be regenerated from that. So we're going to look at the differences. Now, the first one that we've got here is an example of something called competitive inhibition. So this basically decreases the enzymatic activity by binding to the active site of the enzyme. And this happens, usually these inhibitors are what we could call a substrate analog. They look like the substrate. They have similar features to the substrate. The equilibrium expression for the breakdown of the enzyme inhibitor complex, it would be given the same way that we could give an equilibrium expression for most other things. It would be, I think that just to show what that means, right? This is just concentration of enzyme, concentration of inhibitor. If you remember your Chem kinetics here, and then the complex. And so we can use Ks to describe the formation and dissociation of the correct pathway, we could say, our enzyme and substrate, but this also can happen with the inhibitor. All right, and so again, the key feature here is that the active site on the enzyme is being bound to by, in this case, the inhibitor, right, kind of has similar features to that substrate, and so that's why they bind. Okay, how do we identify a competitive inhibitor? It's actually worth noting too, when we're talking about competitive inhibitors, how we can actually overcome the inhibition. Well, competitive inhibitors can be overcome by increasing substrate concentration. If you increase the concentration of the substrate, this will outcompete the inhibitor for the enzyme. And this depends, of course, on the Km. Some substrates will have higher or lower affinities, but in general, any inhibition by an inhibitor can be overcome by adding more substrate. And again, this only works with competitive inhibition. Now, how do we tell if we're looking at competitive inhibition? So in the presence of a competitive inhibitor, the slope of the Lineweaver-Burke plot changes. Okay, so the y-intercept of the graph does not change. So the y-intercept being the Vmax. So 1 over Vmax remains unchanged, but Km actually increases by a certain factor. It increases by a factor of 1 plus inhibitor over Ki. I don't know if I've got that on this graph. Yep, it's right here. So this is the normal equation. And what's happening is we're having to add this term in here. And so, but what we see and what we need to be able to recognize and what you guys need to be able to recognize is that with no inhibitor compared to adding different amounts of inhibitor, we see Vmax remains the same and Km changes. This is proof of a competitive inhibitor. Okay. All right. Let's talk about non-competitive inhibition. So non-competitive inhibition is a form of enzyme inactivation in which the substrate or a substance binds to a site that's other than the active site. So and then it distorts the active site. And so what you can see here is you've got an enzyme. It looks like binding that same circular oval type substrate would be the preferred method, right? It has the option, so there's a little more complicated equilibria going on here, right? We have an equilibrium between the enzyme and the substrate. We also have an equilibrium between the enzyme and the inhibitor. So the enzyme can bind to the inhibitor, and you can see here that that might, of course it doesn't exactly show it changing the active site shape or anything. And then the inhibitor enzyme complex can also become the inhibitor substrate enzyme complex, and vice versa with the enzyme substrate can become the enzyme substrate inhibitor complex. So there's a few equilibria. And then only the enzyme substrate complex, though, can form enzyme product. And so that basically ties up this enzyme in a whole lot of other busy work. So as an effect, it's not making as much product, and so that might make sense that the Vmax is going to change. So here you see Km doesn't change, and of course it wouldn't. The substrate still has the same affinity for the enzyme, still binds. But it inhibits the reaction. So it doesn't inhibit the binding, but it inhibits the reaction. And so we see, again, when looking at a Lineweaver-Burke plot, we see that the Vmax changes. So that's how we spot a non-competitive, a non-competitive inhibitor. The term changes. I don't think that part matters so much. The term for our Vmax back in our previous equation actually gets a little bit bigger for this non-competitive inhibition. We would add this term as well as this term. So this would be for competitive inhibition. We would add that term. Non-competitive inhibition, we're gonna add both terms actually. They both get added. I'm looking at the equation in the book, and it's added to both the slope term and the y-intercept term. Okay. Wait. Yeah. Slope and the y-intercept change. Yeah. Slope and the y-intercept change. That makes sense. The x-intercept doesn't change. That's the Km. Okay. Sorry. Let's see. So again, you should try some of the practice problems in the book that just say, hey, here's a type of inhibitor, what type is it? And then it gives you data. And you should plot it, compare it to the original graph, and see what changes. Does Vmax change? Does Km change? All right, a couple more types of inhibition. First, let's talk about mixed inhibition. So similar to competitive inhibition, sorry, I misspoke. Similar to non-competitive inhibition is mixed non-competitive. Now, mixed non-competitive inhibition does affect the binding of S. And so we do see changes in the Km, but it also, the dissociation constants of Ki, of K inhibitor, and also of activated K inhibitor, which was, I guess, in the last example, or here we can see it as this with a prime, sort of like the activated version. The dissociation constants aren't the same. And so then again, the enzyme's getting tied up being part of different complexes. And so Vmax changes as well. So it affects the binding. So Km changes. It affects the amount of free enzyme or the enzyme substrate complex. So Vmax changes. So with mixed non-competitive, we see both things changing. We see the slope and the Vmax changing, or the slope and the y-intercept, and we also see the Km. Okay. There's one more type of inhibition, and this is uncompetitive. Competitive. Uncompetitive inhibition. And this one is a little funny. So we've got enzyme substrate that can react to form enzyme substrate complex. And the inhibitor can actually bind to the enzyme substrate complex and not to the free enzyme. And so while some of the enzyme substrate complex can go on to make product, we do get some binding to the inhibitor. So like K I minus backwards direction and K inhibitor. And so with this one, the Lineweaver-Burke plot contains parallel lines. We see that Vmax decreases and Km decreases. And so rather than in the mixed non-competitive where they look like they more crisscross, one increases, one decreases, in this case, they both decrease and the line sort of shifts over. So that's how you can spot competitive, non-competitive, mixed non-competitive, and uncompetitive differences. Now, there's also irreversible inhibition. So we can take a second to talk about irreversible. And irreversible, sometimes known as suicide substrates, molecules that can bind irreversibly to an enzyme and basically inactivate it, sometimes called Trojan horse substrates. These are used in medicine all the time, like penicillin is an antibiotic that goes and just binds to bacterial enzymes and just inactivates them. But these are usually covalent binding. So once you covalently bind to the inside of an enzyme's active site or to some other part of it that changes its shape, it's done. It's not going to be useful anymore. Okay, so thanks for staying tuned for this video, guys. I know it was probably a long one. That'll be it for today. We're not going to do more of these videos today. I'll post the second half of the enzyme topic. I think it's regulation and mechanisms tomorrow, and then you'll have your first exam on Friday. All right, guys. Stay safe out there. See you next time.